And today we will cover some important topics on Africa, race and ancestry, environmental data, and importantly, securing future funding opportunities. It's great to have Nikki Mulder here to kick off day two. We want to highlight what's happening in Africa, and in the coming months, we will introduce the IHCC to the African Population Cohorts Consortium, slated for funding later this year or early next year. But in the meantime, it's a pleasure for me to introduce Nikki. She heads the computational biology group at the University of Cape Town, has been leading genomic efforts on the African continent, including her involvement with H3 Africa and H3 BioNet. She's also a member of the IHCC steering committee and has recently won a great award from NIH to support data science research in Africa. And we've asked her this morning to tell us about this initiative and perhaps to forecast how it might work with IHCC. So welcome, Nikki. It's great to have you here today.
Thanks, Jeff. Thanks for the invitation to be here to present some of the work that we're going to be doing. So if I can have the first slide, please. We, as Jeff mentioned, we've recently been awarded a new grant by the NIH. So this is part of the NIH's DSR Africa program. On the next slide, please. So the DSR Africa Consortium is basically a new program to look at the application of data science techniques and technologies to make novel discoveries that have real health benefits. And so they have a big consortium where they're funding research hubs to look at analysis of existing or some new data, some training projects where they're looking at building new data science training programs in Africa, ethical, legal, social projects, and then a big data, open data science platform and coordinating center. Next slide, please. So the projects that have been funded, so these are now available on the NIH website. So there are 7 research hubs that have been funded. I'm not going to talk about each of them, but you can see that there's a very diverse set of topics being covered, from infection dynamics and pandemics to antimicrobial resistance, injury and surgery, impacts of climate change on health, etc. Next slide, please. And then the ELSI projects, there are 4 ELSI projects that are also looking at things related to law and big data in genomic medicine, 7 training grants. And then the award that I've got is for the Open Data Science Platform and Coordinating Center. So the Coordinating Center part is actually, the PI is Michelle Skelton, who is a current Coordinating Center head for the H3 Africa Consortium. So, and then I'm leading the Open Data Science Platform, which I'm going to tell you about now. So on the next slide, the aim of the Open Data Science Platform is to provide a gateway for African researchers who are working in this consortium, who have multiple different data types and want to use data science techniques to basically make these novel discoveries. So we have to provide this big open science platform where they can have their data, they can search metadata, they can bring in tools, they can run workflows directly on different cloud computings. So the platform is called Elwazi, which is basically from a couple of African words, meaning knowledge, robust. And so we're building the Elwazi Open Data Science Platform. Next slide, please. So our first aim is to actually build the infrastructure. And so this is based on the data biosphere concept, which has been around for a little while. It's been used. So it's used in the Terra platform framework and in Gen 3. And it's been used for high-profile projects like the Human Cell Atlas, the All of Us program, etc. So we're using that same structure, and we've got some of those people on board as well. And as I said, the first aim is to build the infrastructure. So what are the different components? We need API access to the platform. We need a tool registry, workflow registry, a data store, and then secure access. So this AAR, the authentication and authorization. So a lot of this draws on a lot of the GF4GH standards. Next slide, please. So the second aim is really to deal with the data that's coming in. So when we put a proposal in, we didn't even know what data types there might be. Now we have a bit of an idea. So we're going to build some metadata models for having standardized data types where there are overlaps in the data elements, have some harmonization procedures and support for that. We might need some reference data sets that multiple different projects need. And so we'll pull those all together into some sort of big metadata model and have a catalog or an atlas that searches that model and enables people to discover these different pieces of data of very different, very diverse types. And then we will also, where necessary, have the actual data sitting in here or on relevant cloud computing environments, or they might be sitting at the research hubs. But all the metadata and everything will be searchable in here, and tools and workflows can be deployed from in here. Next slide, please. So the third, so this is kind of showing this if you just click through. So you have the data coming in on the next click. We have then this public access to, identifying the data types, and then you'll also have user workspaces where individual projects will go in and they'll have access to their own data sets like the Terra platform. And then they'll be able to pull in data from different sites, specifically ones that have been authorized to access that are in this platform. Next slide, please. The third aim is to bring in the tools and the workflows. So again, it was hypothetical when we put the proposal in. We didn't know what tools were going to be required. Obviously, there's some standard AI, machine learning, various data science applications. So we're now engaging with the projects to see exactly what their tool requirements are, building some workflows with them. And then we'll have these interfaced with the various cloud providers. So we're engaging with AWS, with Google Cloud Platform, and with Azure. And then also hoping to have some on-premise facilities for when the data doesn't want to, people don't want to use the cloud, or they have local facilities already that they prefer to use. And so we're building the set of tools and having these registered. Next slide, please. So this is just, if you click through, this is just showing how these different workflows and tools will be pulled into the registry. They'll be indexed in the tool registry, and then you'll have WES, which is the GA4GH execution service, to be able to run the particular workflows over your cloud environment of choice. Next slide. So the idea is to have these different facilities, because in Africa, you can't just say you all have to use AWS. Everybody has their different limitations and what they prefer to use. Some data can't move. And so we're going to have these facilities set up across the continent where people can have local data stores or have access to sort of shared data stores and be able to choose their data and then run their workflows. Next slide. And then, of course, we'll provide a lot of user support on the next slide. This includes for the training grants, having the data, having the training materials made fair and put in the system. For the research hubs, having a nice two interfaces, one for programmatic access and one for your more novice users, where they can have sort of Jupyter notebooks and sort of easy point and click access to tools. And then we'll have a help desk and then additional support like clinical data for REDCap, etc. Next slide. And then finally, we'll be doing some monitoring, outreach, and sustainability. So we'll want to monitor the platform usage and impact, dissemination. So make sure we, everybody knows about it, how to use it, provide support for using the platform, etc. And then ease access to actually using the clouds through the NIH Strides program. So next slide, please. So this is just sort of a summary overview of it. We'll have this gateway where we have access to documentation, protocols, help desk, any training materials, and then the Alwazi, the actual open data science platform. The first instance is going to be based on the Terra system with a customized interface. So users will come in where they need to use on-premise solutions or AWS, then we'll use the Gen 3 framework. But for the user, you'll have a single interface where they can come in within their projects, share the resources, grab the data that they need, find the tools they need, and then run their various analyses on the cloud, of choice, or the computing resource of choice. And we think that this is going to be very scalable to many other resources. So while at the moment there's 7 research hubs that will have their data, their tools, and facilitate access for those groups, it's going to be very flexible to enable other data types. So we've tried to leave the metadata model quite flexible and modular so that we can plug in whether it's environmental, climate, clinical, genomic, metabolomic, proteomic. So we can actually plug in all sorts of different kinds of data types here. And we can have this interfacing, because it's going to be on these existing cloud platforms. It can also interface with these big programs like the All of Us and the Human Cell Atlas. So we hope that this is going to really go a long way towards building an open science platform that is really African specific. So it takes into account the limitations. We've been working in Africa for 9 years. We know what the limitations are. We know what the perceptions are, and we want to tailor this that makes African scientists comfortable using the system, and ease the burden to using cloud facilities. So we hope this will be the start of many other collaborations too. So on the next slide, the final slide, I just want to acknowledge the partners. So the Coordinating Centre PI, as I said, is Dr. Michelle Skelton, and we have a number of partners in Africa, and including some of the GA4GH members and members who've been involved in Gen3 Terra and this biodatasphere concept. So thanks, Jeff.
Great, thank you, Nikki, and congratulations again on getting this impressive award. As the slide just that was just up suggested, please put questions in the chat now or throughout the program today. And I just, with our IHCC hat on, you alluded to the interaction between the El-Wazi platform and all of us and some longitudinal population studies. It seemed like some of the elements in your proposal in terms of training, open science, and policy issues that you're going to address are certainly things that we're talking about here across the IHCC. I'm just wondering if you could comment on how we might work more closely together on very similar types of issues.
Yeah, I see quite a few overlaps. In fact, even the cohorts atlas of the IHCC is something that we might even use, reuse the same code and have the model sort of harmonized such that, if you wanted to, extend our data cohorts to the IHCC ones, that there's sort of ease of doing so. We'll be obviously doing a lot of training in data science, but very specifically to biomedical applications. So I think that overlaps with the IHCC's need to do some training. So we'll need to do training both in data science technologies, but also then in how to use this particular platform. So I can see that we'll have some overlaps there. We're hoping to get as many cohorts on board as possible. So even though this is in the start, it's really for the data science research hubs, but we want to reach out to other funders who've funded cohorts with very diverse data types, because there's going to be an environmental session later today. There'll be new data types for environmental data, for climate change. So we want to have this as a platform that goes beyond just the cohort and its clinical and genomic data, but to other aspects of things that impact health. And because IHCC is also moving into those different spaces, I think it'll be a good opportunity to work very closely with the IHCC to think about how best to model that and how best to integrate that, and then have our different resources interoperable.
Yeah, no, thanks for that answer. I think from a very practical perspective, for one, we don't want to see these open science platforms evolving away from each other over time. So the more on the ground floor we can create those harmonization opportunities, I think the better. And to the extent that it's possible, I think having some of your people involved in training, involved in the open science platform, getting involved with some of our working groups, so that there's that touch point. It's great to have you, but I think also others from your group and across Africa it's a welcome. It's an open invitation for you to be more involved in helping us chart our course and vice versa.
That's fantastic, because I think we will both learn from each other, and we also want to always have a strong capacity development component to our projects, and so getting our African scientists involved in the IHCC activities where there are overlaps will be a great opportunity for us, and I think also for you in terms of being more relevant to different areas.
Well, thanks again, and congratulations. We look forward to charting your progress, or also doing it together, as we just talked about. And again, thanks for being here today. And now I'm going to turn it over to Philip Awadallah, who is chairing the next session on race, ethnicity, and ancestry. Philip?
Good morning, everyone. Good afternoon as well. Happy to be. I'm Philip Awadallah. I'm at the Ontario Institute for Cancer and the University of Toronto. I'll be chairing this next session on race, ethnicity, and ancestry. Today, we're going to have two presentations, a live platform discussion following those presentations as well. And so this next session is going to be, as Jeff had just mentioned, following the session title is called Race, Ethnicity, and Ancestry. We have two presentations, one by Chantalier and the second by Melanie To. Shaniqua Collier is a associate professor at the George Washington University. She's in the Department of at George Washington. She's also a special volunteer at the Center for Research of Genomics and Global Health. And our second presentation is by Melanie Courteau, who many of you know, who's a critical member of the Data Infrastructure Working Group at IHCC. And she's at the EMBL EBI and metadata standards in the Archival Infrastructure and Technology team. So this morning, we're going to start with Shaniqua Collier first. Welcome, Shaniqua. Very happy to have you here.
Good morning. Thank you so much for having me. It's great to be a part of this important discussion. So let's start with my agenda. I was asked to discuss, next slide, please, race, ethnicity, ancestry, and data collection to explain what labels are used in genomics research and why it is important to collect these data in genomics research. I'm speaking from the perspective of someone who has a bioethics and law background and who works in the ELSI, ethical, legal, and social implications space, and who also collaborates a great deal with geneticists. So here is the agenda for the next couple of minutes. Next slide. So let's start with the genomics research landscape. We are witnessing an unprecedented interest globally in diversity, equity, and inclusion in research in response to reports and, frankly, alarm bells that less than 3%, in some cases less than 2.4% of GWAS studies from traditionally excluded and marginalized populations, Latinx, Indigenous, African diaspora populations are included in these studies. So we know that diversity and inclusion is an ethical requirement and also a scientific imperative for us to better understand global genomic diversity. We also know that scientists who enroll diverse populations in research often use broad categories, sometimes called supercategories, which reflect how they perceive a population. For example, we are all familiar with African ancestry populations as a phrase, European ancestry populations. Sometimes geneticists report results using racial categories or ethnic categories. Empirical research has shown that investigators almost never define how data were selected to fit within racial, ethnic, or ancestry categories in publications. There may be an assumption that we all mean the same thing, but these categories change in meaning as we move across the globe and over time. Sometimes such labels are important to study questions and results, but it would be important for investigators to explain what makes these categories important in a particular study and how ancestry is determined. Authors often don't say in their publications. So here we are as a field with these goals. Meanwhile, in the most recent strategic plan, NHGRI stated its aspiration to move beyond what it calls social population descriptors, and race is one of them. This is a clear goal outlined by NHGRI. Less clear is how to achieve this goal. What population descriptors should we use? What happens next when we move beyond race? Next slide, please. So now I would like to talk with you about a survey collaboration with my wonderful colleagues, Dr. Charmaine Royal at Duke University, Yingwei Yang, Shantanu Srivatsa, and also various leaders at IHCCC. This is an IHCC collaboration. And we have discussed exploring what labels, if any, are used when IHCC investigators report into studies. We believe IHCC can help answer these questions and interrogate the prediction or aspiration of NHGRI. We have emailed you, members of our cohort, and we are so grateful that some of you have responded to the survey. Thank you so very much. For everyone else, please let us know what categories are you using. Please respond to the survey. So the survey is still in its beginning data collection phase. I will not be providing formal research results. Next slide, please. But I would like to give you a snapshot of what we are seeing so far for some but not all of the survey questions. So we administered this survey I think at the end of the summer, and we received responses representing 6 countries and 7 cohorts, 2 in the United States. Next image, please. Here are responses to the question: do you collect race, ethnicity, ancestry, tribe, or other descent-related data? And we asked survey respondents to check all that apply. So you can see that there are some differences in terms of the countries where the cohorts come from, some for political reasons, whether they actually collect race, ethnicity, or ancestry data. Next slide, please. Then we asked, please list the categories you use to collect race, ethnicity, ancestry, and descent-related information. And we received responses from a few cohorts. Next image. In Brazil, you can see those categories. Next. Qatar and America, one U.S. cohort following racial categories and ethnic categories. Next, please. Okay, great. So respondents in Brazil and France also listed ancestry categories. In France, for instance, they ask about parents, birthplace, and country as a way of assessing ancestry. Next slide, please. We also asked, do you determine race, ethnicity, or ancestry descent-related information using a genetic assessment, or do you assign ancestry? And we received yes responses from cohorts in Brazil, Qatar, and one US cohort. And so you can see who looks at where we're looking at genetic assessment and where cohorts are actually assigning ancestry to participants in the study. All right, so what is the potential for this? We would really love to hear from as many of our IHCC cohorts and colleagues as possible to better understand how to address the challenges of using population descriptors in research. I'd like to spend a short time explaining some of the challenges that we're thinking about. So one relates to how we use population descriptors. What is the rationale or the circumstances that lead to the categories? And sometimes the rationale and the circumstances are not clear. This is especially true for populations that are less represented in research. So a quick look at this classification system published by NHGRI shows that some African populations are included in uncategorized population and undefined ancestry population. One observation that stands out to me is that there is more specificity here for European populations and genetically isolated populations. Pope, Joy, and Fullerton published in their well-known article, Genomics is Failing on Diversity, that GWAS studies often use Black or African ancestry or sub-Saharan Africa when describing population samples from Africa, and provide more details generally when describing populations from Europe. So as a genomics research community, we might strive for equity and representation, definition, and categorization. A particularly tough question might be, should we develop population labels that local populations are going to buy into, or at least collect metadata on social identities, in addition to other important factors besides social identity, besides genetics? What other factors and data are going to get us closer to understanding health and health outcomes? Next slide, please. Looking toward the future, will our new population descriptors advance or hinder understanding about underlying diversity and ancestry within these population categories? Will these new descriptors play any role at all? Will they erase and marginalize some diverse ancestries, or provide clarity? How can we collectively chart a pathway forward? And so here's a publication by my colleagues that observed 21 ancestries in their study and showed that on average, people had multiple ancestries, at least 4 in the populations that they observed and examined. Next slides, Next slide, please. So what are the harms when continental labels don't capture underlying diversity? Harms include obscuring underlying genetic and ancestry diversity from analysis that could have medical and health implications. So this study has been published for a while now, but I think it provides a really great image to show how there can be medically relevant differences within the pharmacogenetics context when looking at population. So in this image, the Luya, the Maasai, and Yoruba populations have different frequencies of alleles that impact abacavir hypersensitivity. But these would all be called African ancestry populations, or even East African populations when talking about the Luya and the Maasai. There are also significant differences between populations listed here that might be categorized broadly as Asian. Take a look at the Chinese Americans versus the Gujarati Indian Americans. Next slide, please. Before concluding, I'd also like to tee up the discussion point about the complexity of these categories. Race, ethnicity, and ancestry can be avowed, perceived, and or ascribed. The twins pictured here may identify as biracial, but one may be treated as Black and another white when enrolled in a study. Dr. Fulwiley wrote that she is Black in America and white in parts of Africa. In the course of a plane ride, her race could change from Black to mixed if she flies from America to Senegal to France. In Chad, a person may identify socially with one ethnic group, but genetically have significant ancestry reflecting Eurasian migration. So incentivizing understanding of the complexity of these identities is critical to the future of research. So as we make decisions to support data sharing on a global level, can we do so without flattening identities, which is incentivized when people use the OMB census categories in the United States? Even if researchers do not necessarily need to compare based on race, they often believe that in order to get funding, they need to do so according to qualitative research. And finally, my last slide, I'll conclude with by reiterating some of the opportunities for IHCC, understanding practices regarding population descriptors within this global consortium, assess ambiguities and inconsistencies in how cohorts collect, record, and report population data. Referring to an idea that Jeff Ginsburg brought up earlier, how do we grow together and evolve together rather than away from each other, as he said earlier? Contribute to the discourse on global harmonization in ethical and scientifically appropriate ways. I look forward to talking more. Thank you.
Hi, thanks, Reniko. Thanks, everybody, for attending this session. I'm Melanie Corto. I'm at EMBL EBI in Cambridge, UK, and I've had the privilege to work with many of you on data harmonization. On the next slide, please. Thank you. So on the next slide, please, a lot of us have come together and worked in building up the IHCC Cohort Atlas, which you may be familiar with. It's available online right now. You can go to atlas.ihccglobal.org. You can browse the atlas. The atlas is really meant to be a discovery tool at this point in time, and to echo some of the things that Nikki mentioned a little bit earlier, moving forward, build on the discovery to go to data access and retrieval and analysis and so on in the longer term. If you go to the atlas, this is a screenshot of the front page of the atlas. You can see that the central pane lists the cohort for which we have harmonized metadata at this point in time. There's an external link at the moment to external cohort site. And most importantly for this session in particular, the left panel shows the filtering abilities that we have for cohorts. So the idea of harmonizing the data dictionaries and building the atlas was enabling people coming and saying, I want to find cohort that collect information about smoking or specific drug exposure, people who live in the USA, and so on and so on. On the next slide, please. So if we look at the current status in particular for the representation of race and ethnicity, so if you look in the IHCC Atlas, the right-hand side showed you a screenshot of the specific filter category. So race and ethnicity, the third entry here, fits under socio-demographic and economic characteristics. So it's in the same overall bucket as a lot of other attributes such as age, education, family and household structure, birthplace, and so on. Native language, which is also relevant here. And you can see immediately that we don't distinguish between ethnicity and race, because in the cohort that we've worked with in this time, the distinction was not necessarily obvious, or we didn't quite understand how to split those attributes. As Shaniqua presented, based on the survey, and again, thanks to those of you who've filled information in the survey, we know that different elements are collected, but also that under the same labels, people may understand different things. So there can be a diversity of semantic understanding of the labels that are being used for the cohort. For example, interestingly, in France, they don't capture race and ethnicity for legal reasons. Instead, they will use birthplace, or the birthplace of your parents, as a proxy for that information. And that comes with all the caveats of it not being the information recorded and being a proxy for, and so on and so on. On the next slide, please. So what I think is missing at the moment, and Shaniqua is really the domain expert with respect to the semantics of the information, but I think we need to, as a community, and that's probably an opportunity for HCC, we need as a community to clearly understand what we're calling what. When I'm describing ethnicity, what do I mean? Is it self-reported ethnicity? Is it based on your birthplace? Is it, what are you calling ancestry? Is it genetic ancestry? We need to understand which categories are being represented in each cohort, and we need to understand which one are useful for what purpose and how they're going to be used, because that impacts the type of information you want to capture. And there's really, I think, a prospective opportunity for HCC to set up those guidelines and have adoption across communities, so HCC, GFOGH, and so on, to have that global adoption and convergence on the model and semantics of the information. The next slide, please. Another opportunity is on, I think, the training and the documentation. So we know that there is information that's already captured. So there's a retrospective work, which is when we try to annotate the cohort information using the label we define in step 1, we will need to rely on the expertise of cohort owner, you on the call here, to understand how to map the existing label in the information to the existing model. But moving forward, it'll be so much easier probably that if we can agree on that first model and semantic, can we train and document those extensively and propose those as a model moving forward, which would remove the mapping step, which can also be a little bit ambiguous. And the last, I think, opportunity is more on the tooling side. So I'm firmly on the tooling side. And I think it's really critical that whichever way we go, whichever label and definition we decide to adopt, it's really important that when you're in the atlas or you're collecting data and you're analyzing data, there's that ability of going back to the original information. So if you're collecting birthplace as a proxy for race, and I'm getting your data set, I need to understand that that's what happened in the data I've used for my research moving forward. So that ability of tracking the provenance and understanding what happened, which mappings were done, who made them and why, and how I can possibly, revert or change them, I think that's going to be a very useful feature that we need to develop on the technical side. And on the last slide, please, I just want to thank everybody who's contributed to the Atlas building. The model has been developed at EBI. The front end is led by OICR, and we've really had extensive support from HCC, the HCC community, in collecting cohort information and helping us build that model, which we mean to be extensible and extended based on your needs here today.
Fantastic. Those were fantastic presentations, everyone. Thank you very much. I hope you can hear me. If you couldn't before, again, those are fantastic presentations. I'm going to start the ball rolling here with some discussion. And hopefully comments will come up in the Q&A and chat as well from our community here. I'm going to start with a question for you, Shaniqua. Great presentation. And first thing I wanted to ask was following on a comment that you made specifically about population labels. Can they be useful and end up marginalized, or do they become instruments in marginalization directly or indirectly? Was maybe hoping that you might want to comment or expand on that in terms of your thoughts there.
Wonderful. I think there are a number of ways we could think about that. One relates to the abacavir hypersensitivity slide and pharmacogenomics, and using a broad label such as East African or West African or Sub-Saharan African or Asian, and not creating policies that are going to help improve health outcomes for subpopulations within those broad categories. So creating a FDA policy that says we're only going to engage in genetic screening for people of European ancestry before we prescribe abacavir, and not analyzing the data that shows that actually people of Gujarati Indian ancestry are more likely, have a higher allele frequency in their population that's going to respond poorly to abacavir. Another, I think, relates to the conversation that we're starting to see with Indigenous and Native American communities, where some authors and scholars are saying that communities should play a role in how they are defined and discussed in the challenge.
So I think we had, did we lose a feed there from Shaniqua? Melanie, I see you.
Yeah, Shaniqua is frozen on my side as well.
I'm afraid Shaniqua's frozen. Oh, we've lost her. Oh. While we're trying to get Shaniqua back, I'll come back to you, Melanie. Oh, hi, Shaniqua.
I'm sorry, I don't know what happened. What was the last thing you heard me say?
I heard I heard about community engagement.
Okay, so that we can understand how people identify themselves. Okay,
Right.
great.
Yeah. So actually, I'll just, because we did lose you, I'll get you just before I go to my question to, sorry, Melanie. Actually, that's been something of a hot topic here in Canada, is self-identification associated with a community, despite or not recognizing, say, genetic ancestry as well. And I agree, it is critical when we think about this, particularly when we're going to see self-reports that are associated with a particular community, and that will shape and define health outcomes, presumably, for those individuals as well. And they are going to be distinct potentially from genetic ancestries, whether individuals know that. Do you have any thoughts there on how we deal with that in terms of capturing that information with regard to cohorts, portals, and so on?
Is that question for me or for Melanie?
Yeah, sorry, that was for you, Shaniqua. Sorry.
Perfect.
Yeah.
Okay. You know, some of our colleagues in South Africa have written papers about collecting metadata on how communities identify themselves, as well as ethical issues related to genomics research. And I'm really interested in that discussion, and if it's possible to further that discussion. There have been some papers on how metadata can play a role in helping us with our data analyses.
Super, thank you. Melanie, you mentioned guidelines, right? And developing guidelines, the importance of developing guidelines. What currently exists, particularly that might be useful from the IHCC perspective as well, given the varied jurisdictions, regions, countries, et cetera, that we are exploring and hoping to develop harmonization activities across the IHCC?
Yeah, thanks, Lee. That's a great question. So there's different efforts, that Shaniqua mentioned. There's different. We know that it's a topic that's becoming increasingly important because it has really impact on health and treatment. So there's several groups have worked on different efforts. So there's the OLS ancestral ontology that Shaniqua presented, which is work done by my colleague at EBI in the GWAS catalog. I know that the paper that Shaniqua referenced on metadata in Africa, I believe Nikki is heavily involved on that effort, and we're looking into an ethno-linguistic ontology, because in Africa it seems that a lot of the categories can be based on the language that the population speak. But we know that that's not true everywhere. So we had the Qatar cohort representative, for example, mentioned that in Qatar, the language is fairly homogeneous, but the populations are not. So I think there's different efforts that are all relevant in the context they're in. And I think the problem and the opportunity at the same time for HCC is how do we bring that globally? And I don't think there's a good answer right now. And that's why I was thinking, I think the first step is understanding what we're doing, where and how, capturing that as accurately as we can, and then move forward and try to see can we drive those guidelines, those global guidelines. But I don't know if it's possible. I hope it is.
Thanks, Melanie. That is helpful. I think I'm going to go to some comments and questions that I'm starting to see arrive. This is a comment, and I think I'm going to ask Melanie, you first to apply to respond to this. This is from Paolo Lutufo. We need more information from surveys related to open questions about race. Census and health surveys apply predetermined categories and can reduce the meaning of race as a social construct. And then he's got an example here from northern Brazil. A large proportion of the population self-declared as brunette. No white, no black, no indigenous. So that I think speaks to how we implement some of these tools, as well as collect them and try to bring them together. I'll start with you, Melanie, and then maybe Shaniqua, if you would like to respond there as well.
Sure, I think that's a very accurate, observation from Paolo. If you remember the slide that Shaniqua was showing with the survey census categories as well, there's no definition there, right? It's going to say white, black, Asian, Hispanic. It's just a label. So different people will understand different things based on that label. For example, in England, we have white British and other white. So it's like, is that the same white than white American, or is that a different type of white? There's no explanation of what that means. Or if there is, sometimes there is published in a guideline, in a PDF buried somewhere, but it's not available to the person who actually fills in the survey at the time at which the data is collected either. So I think there's a need to have a census of all those categories. For example, brunette that Paulo mentions, and understanding what it means. It sounds based on what it's describing that it's a self-reported type of categories, but understanding what that means and in which context.
Great, thanks, Melanie. There's a question here from Pabalo. Thank you, Melanie, for the presentation. How do you communicate research in a mindful and culturally sensitive way to avoid stigmatization of certain populations? I just saw on your slides that filters did not differentiate or pick up race and ethnicity, and was a bit concerned that could add to the stigma most of the scientific world is trying to fight.
Thanks, Pablo. I think on the race ethnicity, and we had a lot of discussion with the Atlas team when we, made that choice. I think we kind of took the possibly easy way out. We thought that bundling it all together meant we were not committing to any interpretation, with the understanding that this is not good enough, but also keeping in mind that we don't know right now how to make it better. And I think the model itself that we're using can be extended. We can split those. We don't know right now how to do it. So we're relying on information from the cohort for the survey, information from experts like Shaniqua and her team to understand how shall we better represent that. If you have any feedback, how we could, as a first step, move forward, we can implement that as well.
Great, thank you very much. Shaniqua, I'm going to refer this question to you from Michelle Ramsey. There are very different approaches in different continents. Place of birth seems problematic, considering the increasing mobility of people. And the same would go for language. So again, second, third generation Americans in Africa, language is often important as a way of thinking about ethnicity as well. So, I mean, that's a combination of both a comment and I think a question as well. So I think to some extent, that's a question of how do we capture that information, and what is its importance as well.
As a person with a bioethics and policy background, I'm really interested in how we capture information and also how we communicate it to non-experts. Given our history of genetics, given eugenics history, given how race, so much of what race is also what it's not, right? It was used to create hierarchies of populations originally. So how do we break away from that past and move forward to something that is meaningful? And how do we do so globally? It's incredibly problematic to me that some researchers export United States OMB census categories for products that they're seeking FDA approval for and apply those categories overseas. We are uniquely positioned as a cohort to come together and address these questions in ethically sensitive and rigorous ways. And I'm really excited about that.
Fantastic. Thank you very much. That last point I was just thinking about in the context of how we capture information and what we call now newcomers, as being just as critical, regardless of ethnicity, let's say, and race, particularly when we're thinking about how some of these activities, these population cohorts and so on. Sorry, my family's just starting to wake up here, so I'm about to be invaded. And how we think and use newcomers, particularly given population cohorts, are often used as surveillance tools. Thinking about this in the context of COVID-19, population cohorts are not the best tool for surveillance mechanisms. Sorry about this. But nonetheless, capturing information about newcomers is just as critical, because what we're seeing there in the context of COVID-19 is that those are the communities that are being impacted perhaps more significantly than other communities. So just thinking about the context of newcomers, how we capture that information and how we communicate that information to other researchers and decision makers, I think is critical, particularly such that we don't marginalize those individuals. Can you go?
I completely agree. And of course, it's important to think about how we can record and capture information about racism, discrimination between continental populations. If we're interested, if our research questions have to do with the role of social stigma and social discrimination, then it's also important to consider when such categories are relevant.
Great, thank you very much. And I'm seeing some other questions coming up here. Jeff is asking, are we ready to include sequence-based ancestry in our data? And what do we do when the sequence and self-report differ? I think that's a question for both you and me, Melanie. Melanie, I'll start with you there. And actually, that might actually come back to you, Chinico, as well. But let's come back. We'll start with you, Melanie. So.
That's a good and hard question. I think we need to be able to capture anything that is relevant now and will be relevant in the future. So to your previous point, Philip, on newcomers, I think an important aspect to consider is that model evolve and categories evolve and information changes. And the type of information we need to capture evolves through time as well. I think that we do need to capture both myself, and I think what you do when they differ depends on the context of the study you're trying to run. So depending on what you're trying to do, if you're looking, at socioeconomic factors and their impact, for example, for COVID pandemic, do people, self-harm more, or things like that, you're probably more interested in the neighborhoods people live in and the level of education and things like that. If you're looking at genetic treatments, you're probably more interested in the genetic ancestry. So I think there's an importance of the context here as well.
Yeah, I'll just add to that. I think too is each of the individual cohorts, as they're collecting genomic or genotypic data, will use that to some extent as self-declared ancestry as almost part of the QC, and to ensure that the quality of the data, and to some extent also we use that sometimes to understand if there's been mix-ups in the lab, biobanking and so on, and so it adds to LIMS, but again, that might not be again, thinking about this from a QC perspective or a biobanking perspective, it may not be pointing to an error in the biobank. It might be pointing to something that's being self-reported differently by an individual. So, for example, somebody might be thinking that they have some percentage indigenous, but they may not actually. But they grew up in a community, a Métis community, for example. Okay, thank you very much. I'm just going through. So, here's a question from Cindy Lawley. How might the IHCC influence the strategy for prioritizing collecting proteomics and metabolomics data for cohorts in a way that integrates lessons learned from the paucity of underrepresented ancestries in the genetic data? I'll start with you, Shaniqua, and then maybe we'll move that over to Melanie. I think that's a question for almost everyone to some extent as well. But Shaniqua, if you have some thoughts there.
My initial response to this question relates to the prior question. I was thinking that it's important to collect different data types so that we can have access to data for the future research questions, just as Melanie suggested. But are we overemphasizing genetics, race, and ancestry? This is a cohort that also looks at environment. When educational attainment is an influencer on health and health outcomes, or access to water, are we asking the questions to bring in all of these other different types of variables too? So and Charmaine and I have talked about this a lot. How do we collect all kinds of data and also tailor our study questions, choose the data that we need to appropriately answer the study questions, and not let what happens in the United States happen globally, where we're constantly always just comparing ancestry and genetics and missing opportunities to look at the role of other variables closely.
Thanks. Melanie, I'm going to take that same question to you as well, if you don't mind.
Yeah, I really like that question, actually. I think it's a very relevant question. So I think the first step, which maybe optimistically I think the community has taken, is we have this discussion. We have those discussion and we're considering those issues. I think that one of the way that I'm thinking about integrating lesson learned, and to go back to Shaniqua's point, I think there's more than just race and ethnicity and ancestry. I think the way that we can possibly learn as a community is having something that I'm personally interested in working on, is having those statistical representation of cohort phenotypes, where we'd be able to say, this cohort has those percentage in each of those categories, whether it's education level, access to water, race, ethnicity, ancestry, and we could compare that across cohort, and that would give us, that would show which cohorts are positioned where, depending on what you're looking for. So if you see that you're looking for race and ethnicity, and you see that the diversity across IHCC cohort is really poor, I think that can be an alarm signal saying, well, what are you guys doing? I think that's an opportunity. If we understand better the type of information we're collecting, we can then start having those analysis at scale globally again, where we understand what's being collected and what is not, and what's not. Why is it not? Is it because we're not involving the right populations? Is it because we're focusing only on white Europeans? And I think that's how we will identify some of those issues.
Great, thank you. And I think all of these questions are extremely important, particularly in the context if we're thinking about gene and environment interactions or ethnicity and environment interactions, even in the context, or especially in the context of global climate change or environmental change, which is going to be impacting certain communities and groups differently than others. And so as we move forward in some of that type of research, having this type of information is going to be ever more critical. Jeff had a, oh, there it is. How can IHCC, sorry, this question from Jeff again. How can IHCC train our cohort leaders and scientists to better understand current bias in our data? Shaniqua, that's specifically to you, I think. Yes.
Well, this is a great start, isn't it? That just having these conversations and to be able to talk with one another, frankly, in different time across time zones about these issues. I think the survey will help us too, as Melanie was saying, to get us started in understanding where we are so that we can begin to think about where we need to go and actually identify examples of what could be representative of bias through training.
I think that's a fantastic point. I think this survey and this entire IHCC activity is going to highlight not only variability in terms of how the cohorts do their work, but even how different regions capture information at the national level or the regional level in terms of census, for example. Even within some place, I can speak specifically to my own environment here in Canada, we see extraordinary variability across provinces from Quebec to Ontario and so on in terms of how people classify or want to categorize individuals. Coming back to your first point, Shaniqua, as well as why do we categorize individuals to some extent as well, it speaks to some sort of bias too. Sorry, go ahead, Shaniqua. Yes.
Can I add a point too? I'm really excited about these programs such as H3 Africa and the data science program that was described earlier, because it's giving researchers investigators who have not been represented in research the chance to really weigh in on this and to teach us something about how they're developing categories, describing and reporting on populations, and identifying and addressing biases within Africa. I think the contributions from these projects will also be really important to our efforts globally to understand and address bias and other challenges.
Shaniqua, I'm gonna raise a point that came up in my discussions recently here, particularly with working with our Indigenous communities, and I'd love to hear your take on this. We recently had a conversation with a national Indigenous coordinating center, let's say, and they were first asking about what information we had on the Indigenous communities in our population cohorts here, and their first question was, well, what gave you the right to even ask about their Indigenous ancestry? And my response, I don't think this was the best response, but I'd like to take your topic, was, well, if I didn't ask about that, the issue that would arise there was that we were intentionally leaving some of these communities behind, particularly in personalized medicine and genetics. Care to comment?
No, I think that's an important point. And I said earlier, I work a lot closely with geneticists, and I actually just had this conversation with a group of geneticists last week on how important it is to advance genomics research to include people from Indigenous populations. And if populations representatives discuss genomic sovereignty, and we're seeing a lot of discussion about that now, we've seen it for a while. My colleagues have talked about who represents the community, who gets to decide who should participate in research, and what happens when a group or population is excluded to our data. But I think the other side of the coin is this idea of collective visioning about the future and engagement to better include people, these different populations, and how the research is conducted and what questions are asked. Because if we want to look toward the future and we want an inclusive world of genomics, data sharing and engagement in the future, I think we have to take steps for that collective work now. To say that as genomicists, we have this goal to understand population genomic diversity, and we're going to get data through the back door however we can, I think jeopardizes the future of trust and global research. And as scientists who have an obligation to produce this great research and to think about fairness and justice, having these conversations with communities to work out how we can go in through the front door, I think is really very important.
I'll take the last point here too before we close this session, and just to follow on your point as well, as a lot of these cohorts that are part of the IHCC have been around for quite some time, in fact, maybe 10 or even 15 years. And I think re-engagement now is critical, just as might have been as critical at baseline, is also just as critical now as we keep moving forward as well. Such that we're using both the data that we've already captured as well as the data that we want to capture is used in a sensitive and obviously in a way that's going to support as many communities as we can.
Can I say one more thing too?
Sure, of course.
The diversity of the workforce. We are seeing, we all know of Nanaba Garrison, for example, a person with expertise in genetics and bioethics who's playing a critical role in creating policies and moving the discussion forward. More diversity and inclusion at the leadership level, I think, could also have a tremendous impact on these kinds of conversations, not just engaging communities for recruitment, but providing funding and publishing opportunities to scientists from these communities too.
Fantastic. Thank you very much, Dr. Callier and Dr. Courteau. It was a real pleasure this morning and this afternoon. And so I'll be ending the session now as we move into our next session. Again, thanks, and it was a fascinating discussion.
My name is Dr. Kyle Messier. I'm a tenure track investigator in the division of the National Toxicology Program. It's at the National Institutes of Environmental Health Sciences in Durham, North Carolina. I have a background in methods and applications of geospatial exposure and risk assessment. And today I will be chairing this session on environment and climate data. And we have two excellent speakers. So our first speaker is Dr. Alison Motsinger-Rife. She is the chief and a principal investigator in the biostatistics and computational biology branch at the National Institutes of Environmental Health Sciences. Dr. Motsinger-Rife earned both an MS in applied statistics and a PhD in human genetics from Vanderbilt University. She was a faculty member at North Carolina State University before joining NIHS as the branch chief. And she has, her research focuses on gene and environment interactions and methods in statistical genetics. Our second speaker will be Dr. Peter Diggle, who is a distinguished university professor of statistics in the Faculty of Health and Medicine at Lancaster University. He also holds a couple adjunct positions in the United States at Johns Hopkins and Yale University. And he was the president of the Royal Statistical Society between 2014 and 2016. He was also co-authored many books in spatial statistics. In fact, one of his books was a key reference for my master's and PhD training in geostatistics. So very, very excited and happy to have both of these speakers here. So our first speaker, Dr. Alison Motsinger-Rife, will be discussing her work at NIHS. Alison.
I'm so grateful to organizers and participants here at IHCC for giving me the opportunity to speak. If you'll advance slides. So I'm going to talk first a little bit about some efforts that are ongoing at NIEHS that hopefully will provide an example of the kinds of work you can do in gene-environment interaction when you have both genetic data and environmental exposure data. So I'm going to give a quick vignette of the types of questions you can ask and answer. And then since this is a workshop, I'm hoping to get some feedback from folks here about some efforts ongoing at NIEHS that I think would really complement efforts here at IHCC. So I'm hoping to get some feedback on the types of environmental data that would be useful and corresponding tools to help people do similar sort of investigations. Next slide. Like I said, I think I'm preaching to the choir here, that sort of we're understanding that complex traits are the results of genes, environments, and their interactions. And we've heard a lot of discussion already in the workshop yesterday and today about how important and difficult collecting environmental data is. Sort of the exposome is a hot topic right now in general. Where sort of people are trying to collect sort of eventually the totality of a person's lifetime exposures, though that is admittedly an impossible goal. And I want to talk briefly about the PEGS cohort that I co-lead at NIEHS. It's the Personalized Environment and Genes Study, with one sort of vignette of the kinds of things we can do with both genes and environment. Next slide. So PEGS is a North Carolina-based cohort where we have a number of different data types that as a statistician, computational biologist are exciting to think about integrating. So we have whole genome sequencing on this cohort. So we've got about 19,000 people enrolled in total. For about 5,000 people, we have whole genome sequencing available. We also have extensive phenotype and health information, first from surveys on about 9,000 participants, and in ongoing efforts. By this summer, we will have electronic health record information for our participants through both Duke and UNC Chapel Hill medical systems that are local. And then we have a number of environmental exposures collected on individuals. We have extensive surveys on about 9,000 individuals, and we are starting to link all of these participants to geospatial exposures, and Kyle that introduced us here is a huge part of that effort. This is an active and ongoing cohort, so we have the opportunity to call participants back in to collect more data, to do more in-depth phenotyping, to really follow up sort of the hypotheses that come from our large data efforts. Next slide. So the environmental data we have right now are, we have 3 large surveys. The first one we call our health and exposure survey, where we've got information on health. Have you been diagnosed of X, Y, and Z? Family history of diseases, questions on occupational exposure, socioeconomic status, lifestyle data. And then we have 2 exposome surveys that try to get at external and external exposures, including medications, physical activity, stress, sleep, diet, chemical, environmental exposures at home and at work. And then have information on sort of core medical history that's collected in the clinical research unit on campus at NIEHS. Next slide. So really briefly, I'll just go over this. Sort of our exposome survey is broken up into 2 parts. We've got about 200 questions on external exposome, characteristics of past and current homes, workplace characteristics, chemical and metal exposures at work, hobby exposures, UV. And then part B is the internal exposome. It's about 240 questions on medication, vitamins, past chemotherapy, physical activity, stress, infections, sleep, diet, and other aspects of sort of the exposome. Next slide. And then we have a growing number of GIS exposure estimates that we're linking. We've collected addresses and address history, including longest lived childhood address, that allows us to link sort of residential history to a number of available geospatial exposure databases, starting with things like distance to airports, caged animal feeding operations, cell towers, drinking water, and are now including other databases with air pollution, groundwater. It's an ongoing list that I'll talk about in more detail a little bit later. Next slide. And I want to show you just really quickly the type of study that we can do when we've collected jointly these kinds of data. So we're recently completed and are getting ready to submit a manuscript where we hypothesize that the proximity to swine caged animal feeding operations, which is a a huge concern in North Carolina specifically, that we hypothesized that proximity to these CAFOs is associated with immune-mediated disorder risk, and that sort of we were looking for both genetic risk factors of immune-mediated diseases, environmental risk factors of immune-mediated diseases, and G by E environment, G by E interactions. Next slide. So we tested first sort of for environmental interactions. We looked at the exposure between CAFO proximity and immune-mediated diseases. I know this is a little bit small and you're seeing a lot of associations here, but we looked at a number of immune-mediated diseases, allergies, asthma, rheumatoid arthritis, the sort of SLE, you'll see a number of them there. And so we saw strong associations of distance to CAFOS operations, and both individually, the immune-mediated disorders, and globally in sort of any, a phenotype of any immune-mediated disorder, showing that there's sort of shared risk factors across the diseases of this type. Next slide. We also tested for genetic association. In this case, we looked at SNPs in genes that had previously been associated with immune-mediated disorders from a GWAS in an external study. And we saw strong associations in the PEGS population with AHR and decreased odds of IMD, any allergy, and seasonal allergy. And we see sort of consistent genetic associations across IMD phenotypes, suggesting common genetic mechanisms. Next slide. And we saw, again, looking at these sort of candidate genetic variants by distance to CAFOS interactions, we saw evidence and strong statistical significance for a number of gene environment interactions. We saw evidence of sort of additive risk G by E, of dominant risk G by E's, recessive, and then very complex gene environment interactions. So this is really exciting to me to be able to see, really get an understanding of sort of what is the genetic risk, what is environmental risk, and then how do those two types interact to try to understand disease. Next slide. And in PEGS, you can imagine there are a number of things that are ongoing. We are looking across not just immune-mediated disorders, but we have, this is just a population-based cohort. So we've got common diseases that are common in the population. So we're doing work in type 2 diabetes, in asthma, and sort of an array of common complex diseases. We're doing exposome-wide association studies, doing things that are familiar to this audience with polygenic risk score development, and then PRS by E interaction. I'm doing some work in sort of pure statistical genetics methods development for prioritizing SNPs for G by E interaction. And I'll point you to a recently developed database that has gone live out of NIEHS that gives you candidate genes for gene environment interrogation across diseases. We're doing GWAS and sort of structural variant and rare variant association analysis, and building our sort of compendium of geospatial exposures. And then linkage to the health records from Duke and UNC Chapel Hill will give us a longitudinal aspect to the cohort that I'm excited about. Next slide. So now we'll switch gears a little bit. So hopefully that gives you just a really brief overview of the kinds of things you can do when you've got both of those sort of multiple types of data sets. But there are also a number of ongoing discussions at NIEHS and several working groups on sort of, when we say environment, and we are NIEHS, if we can't sort of articulate what we mean by environment, nobody's got a shot. But talking a lot about, so if we want to collect exposome data, what are sources of that data? So that can come from biomarker measurements. So I know some of the cohorts here in IHCC may have biobanked blood samples, urine, plasma, saliva, feces, placenta, umbilical cord, breast milk, hair, or nail sort of samples. And with sort of improvements in technology and mass spec and other sort of aspects, can directly measure environmental exposures in some of those biospecimens. Next slide, please. Also, sort of questionnaires are widely established, though not sort of widely established, not easily validated, where there are all sorts of information you can collect on residential exposures, occupational exposures, socioeconomic status, education, diet, lifestyle, medications. And there are ongoing efforts on campus to really try to develop as generalizable a set of exposure questions as possible. And I guess I am directly asking for sort of input from folks here on IHCC on how we make those questionnaires globally accessible. For example, we started some draft questionnaires to propose to the All of Us effort to incorporate that come from things like the Phoenix Toolkit that have done some measures of validation across different countries, but certainly have not been validated globally, have not yet been translated into multiple languages. These efforts are at the start, but I think would really love to work with IHCC on trying to mature that. Next slide. And then I mentioned earlier and showed you the CAFOS example of geospatial exposures can be a great source of sort of exposome data. And in some ways are the, in my mind, one of the more easily translatable sort of types of exposures. So any cohort that has addresses could link to these resources, and I'll talk in more detail about what some of those resources actually are. So any cohort that's got residential addresses, address history, hopefully maybe even occupational childhood addresses, could be linked with the proper tools to these geospatial exposures. Next slide. Like I said, there are data resources with exposure models and sort of wearables and smartphones. Sorry, I've got a little bit of duplicate information here. Next slide. So there are a number of these sort of both types of geospatial exposures. There are different models and sources of those models and databases that are available at different sort of global levels. There's some that I'll discuss further that are available globally, where others are even local to North Carolina as I think about the PEGS study. So we can think about pulling in data on weather and atmosphere, on air pollution, groundwater contamination, distance to hazards, health disparities information, and built environment, green space, that sort of thing. Next slide. But one of the bigger challenges there is really in munging that data. So like I mentioned, these databases have different geographic extents, different data sources, different spatial resolutions, different temporal resolutions, and they're not consistently structured. And there is a real large activation energy in being able to pull some of those data resources together. And so there's some ongoing efforts I wanted to mention and get feedback on to help make that easier for folks. Next slide. Like I said, we've got efforts that are ongoing and just ramping up, including the addition of someone we're calling a data scholar that starts next week, that's going to work to build software resources to empower assembling those geospatial exposures readily. Next slide. I know this is too small to see, but wanted to put it on here of we're starting with a list of what some of those exposures actually are, what sort of resources are out there. And I can share this in more readable format, but we've started with here are resources that are globally available, here are resources that are available first, we started with the United States, given our location and bias. And this list even includes things that are North Carolina based with our sort of my personal interest in PEGS. And we have pulled together and have tools and code already to pull some of these together, but would love to hear what would be of most interest and most impact globally as we look to start building out the software tools that we hope to disseminate. And we will work closely with the sort of the data working groups here in IHCC. Next slide. So really, within the survey that recently went out about for IHCC in general. You'll notice we've included some questions on environmental data, both what you have collected as well as what would be of interest. And I'm hoping in this workshop we can start some of that discussion. So sort of we immediately can think of prioritizing any of the sort of linkages to databases that are global as sort of highest impact. But even within that list, would be interested in what people find most relevant for their work. So I'm interested in feedback of, is lifestyle sort of the most relevant to your cohort? Built environment, is weather and atmosphere something that you think is interesting and would immediately tie into climate change kind of efforts? Stress, is it toxic and chemical exposure, diet and medication, social determinants of health, others? I'm really interested in hearing what folks would like to prioritize. Next slide. I want to give quick acknowledgements to the number of people that are working on these efforts at NIEHS, both in the PEGS leadership, the GIS modeling, and sort of the common disease working group that is folks from the Division of Intramural Research, as well as folks in DIRT that do the sort of grant funding arm, and even sort of the Office of the Director at campus has been really fun and rewarding as we work to sort of think to learn what the community needs, and as we think about trying to build infrastructure and tools to meet those needs. Next slide. So yeah, I'll stop here. I know in some ways us being limited to chat here is a little bit difficult for discussion, but yeah, I'm asking for input in some of those topics. So if you're willing to share what would be priority, that would be great. I'll stop here and let Peter take it away.
Okay, very nice to be here. Thanks, Alison, for that talk. Next slide, please. What I'm going to do in the next few minutes is just give you a bit of a headline for a report that's going to come out later this year from the Wellcome Trust that I co-authored with a climate scientist, Hannah Neeson, and a biostatistical colleague at Lancaster, Claudio Frontera. And the first thing that we found when we started looking at this report, which was about the potential for using health cohort studies in conjunction with climate data to understand climate health interactions. Next slide, please. Was the almost perfect mismatch between the typical data formats that you get in those two spheres. So it's a generalization, there are exceptions, but broadly speaking, longitudinal population health studies tend to involve large numbers of individuals, but a small number of follow-up times on each. Typically annually or often longer gaps between intervals, but you collect, as Alison has demonstrated, very many variables on each person that's included in the study. And they tend, again, it's a generalization, to have a fairly tight geographical span. In the UK, there are several local cohort studies, for example, centered in particular parts of the country. Now, in contrast, climate data, they tend to work on a rather sparse network of monitoring sites, at each of which they collect very long time series at high frequency, often hourly, or even sometimes more frequently than that. But what they measure tends to be just a very small number of key climatic variables, like notably temperature and precipitation. And because they're based on networks of monitoring sites, they're often geographically very wide in their span, but correspondingly sparse in that you don't have many monitoring sites within a particular district, or even sometimes within a particular country. Next slide, please. So I'm going to mention 4 challenges and then suggest that they collectively present a huge opportunity for IHCC. So the interdisciplinary challenge, the obvious part of this diagram is on the left, where if you want to understand the causal impact of climate on health, then you've got a relatively simple system to understand. Climate pointing into health, and you can build your predictive models and validate them. Life gets a bit complicated, though, when you add society to the mix, because at the top of this diagram, the way the climate changes is going to affect the way society behaves and evolves, but conversely, there's feedback because the way society behaves, most notably developed societies pumping out industrial pollution, affects how the climate behaves. Similarly, the way people behave clearly has an impact on their health. And there should be another reverse arrow there because the way public health evolves has implications for behavior, most obviously and dramatically in the recent COVID and ongoing COVID epidemic, which has changed behavior profoundly, although probably, sadly, in some respects, temporarily. Next slide, please. So the second challenge is the geographical challenge. And this map here shows what we found in our report as a reasonable representation of the coverage of different parts of the world by longitudinal population health studies. And the most striking feature of this map is the relative sparsity of coverage of the African continent. And I know that's something that IHCC are actively working to redress, along with, as Jeff Ginsburg mentioned in his introduction, the successful bidders for a welcome call for an Africa-wide population cohort consortium. Next slide, please. Now, this is the design challenge. This might take a minute or two to explain the background. Rongelap Island is part of the Republic of the Marshall Islands in the Pacific Ocean, and it was contaminated by nuclear testing in 1954. And you can see from the picture there that at the time, it was a well-established community with a church and houses and shops and people earning their living in the way that people do. The islanders, however, suffered various health problems from nuclear fallout contamination following that nuclear testing program. And to make a long story short, the community was evacuated by Greenpeace in 1985. But in 1997, studies were initiated to estimate the residual pattern of contamination of the island to see whether it was suitable for rehabilitation. And the picture on the left shows you the study design that was conducted in 2 phases. The initial study were the open circles, which you can see were spread evenly across the island. And having then established that there was some contrasting pattern in the western end and the eastern end, where respectively people live and people grow their crops, I advised the study team to go in for a second sampling effort focused on the blue points, which, as you can see, are much more closely spaced than the original points. And the point here is that it's not enough in situations like this to just estimate an average. If you're living on this island, you care about the extremes of contamination as much as you care, more than you can care about the average. And so you need to understand the spatial scales of variation to see how finely you need to sample in order to get an accurate handle on those extremes. And I think the same is true of climate exposure, that it's not the average climate that causes the most damage, it's the variations about the average and the extremes, and you need to capture those in suitable metrics, and you need suitable experimental designs in order to make sure that your data are capable of capturing those metrics. Next slide, please. And then we have the analysis challenge. And the key here is data synthesis. And in the opening talk this morning from Cape Town, South Africa, we had a very good example of the need to synthesize information from multiple data sources. But the schematic on the right, which is a cartoon, it's not real data, is intended to convey the flavor of what I mean by data synthesis in the context of geospatial analysis of health outcomes. So we have a political background here. In this case, it's whole countries. If you zoom in, you'd see individual districts. And there will be data that's presented as characteristics of those geographical areas. They might reflect different national policies in different countries. They might reflect different local policies if you subdivided each country into districts. And that's the partition into DRC, Cameroon, and the various other countries in this part of Africa. If you look at the shaded circles, you can imagine there that you've actually gone in to particular communities within those countries and you've measured an outcome. It might be, for example, the prevalence of a particular disease, and the color shading, the gray shading, shows you that you have high prevalence in some of those communities, dark grays, and much lower prevalence in others. The individual red dots are meant to indicate that in some contexts, what you actually get are individual case reports. This is particularly true of rare diseases, of course, and they represent individual points in a spatial continuum. And then finally, the grid that overlays that map is meant to symbolize the fact that we have information on various climatic and other environmental exposures available to us in the form of digital images, pixelated to various degrees of resolution according, again, to context. And so the challenge for somebody like me, who's a statistician, is to say, what's the efficient way to combine all of that information? And this is actually, it may surprise you to know that this is a topic of considerable current statistical research interest, because the traditional way to do it is to massage those various data sources into some kind of common format so that you can line them up in a spreadsheet and do conventional regression analysis of the combined data sets. But the way that statisticians are now thinking is more, if you look at the bottom here, I'll take you through this very limited piece of algebra, is to think first not about your data, but about a single scientific process, P, that you're trying to understand. And then to think of each of your data sources, D1 up to DM, as different ways of learning something about that common process. And so what you then do is you synthesize the information not by massaging the data into a common format, but by using Bayes' theorem to say that if I can have a scientific model for my process, P, and if I have statistical models for how each of my data sets individually relates to that process, so I have D1 conditional on P, D2 conditional on P, dm conditional on P. Bayes' theorem tells me how to convert that into a statement about the process of interest conditional on all of the data jointly. And so modeling in this hierarchical way, by modeling a process and modeling various data streams conditional on that process, is, according to contemporary statistical thinking, a much better way to proceed than trying to massage all your data into a common format to give you a simple classical statistical analysis. And this is particularly the case when, as I think is almost universally true in the climate health space, you are dealing with data sources that show spatial and temporal correlation. And so you need to model those spatial and temporal processes. You can't be using methods that treat the data just as an independent random sample from a distribution. Next slide, please. Okay, well, turning challenges into opportunities, and I want to particularly be a little bit bold and suggest that the IHCC is very well placed to follow this chain of opportunities, and that it's a very good arrangement for getting people from different disciplines to work together. So it certainly has an opportunity to respond to the interdisciplinarity challenge. It also has an opportunity to respond to the geographical challenge, because I know from conversations with Jeff Ginsburg that there are active plans to increase the penetration of IHCC, or the membership of IHCC, into, in particular, the African continent and other low and middle income areas of the world which are currently underrepresented. And in that expansion, because you can make a virtue of the fact that you don't have well-established existing cohorts, because you can design your studies to actually efficiently collect data that will best address the questions you're interested in at the climate health interface. Now, you may be interested in other things, and that's absolutely fine, but my particular pitch today is to try to encourage IHCC to think of what it can do at the climate health interface, and the opportunity to design studies in virgin territory rather than have to retrofit your population health studies to climate research questions, I think is a great opportunity. And then finally, you have the opportunity to work with state-of-the-art statistical methods so that you actually get the best possible information from your hard-won data. Next slide, please. Now, in the report, we actually try to address the question of how should Wellcome, in particular, who are the customers for our report, respond to the challenges presented by the need to synthesize longitudinal population health studies with climate data. And in a sort of increasing scale of ambition, you could say, well, really, we just need to sort of look at existing data and put it together. Moving up the scale of ambition, we might want to actually modify existing longitudinal population studies so that they're better suited to interacting with climate science. We might want to fund new studies ab initio. We certainly want to consider looking at consortia rather than single studies in order to get the geographical spread that we need if we want to understand climate effects on health. And we might even want, as welcome, to set up some sort of global scale center of excellence, which focuses specifically on research at the climate health interface. Next slide, please. But finally, and again, thinking specifically of opportunities for IHCC, we put in a whole set of recommendations in our report. And I'd like to just focus attentions on the first and the last of these. So if the first one is that there clearly is opportunity here for existing IHCC studies to be melded with existing climate research resources and climate data resources to actually start doing some secondary analysis informed by really state-of-the-art and in some cases new statistical methodologies to really get a better understanding of the various drivers in the climate health pathways. And then moving down to the bottom, we made a very explicit recommendation that Wellcome might want to commission selected longitudinal population study consortia to consider how they could reorient some of their work towards climate health research. And we name-checked IHCC and to give you a fair balanced picture, we name-checked IHCC and the Prospective African Population Cohorts Consortium as prime candidates that might be able to respond in this way. The report itself, if I could go very briefly back to the first slide. Apparently not, no worries, it's not important. The report itself will be published by Wellcome towards the end of the year, and we're very much hoping that it'll generate some interest amongst the climate health research communities, and more specifically will generate some decisions from the Wellcome board to actually make some new and major investments in this, I hope you'll all agree, increasingly important area. Thank you very much.
All right. Thank you, Peter and Allison. In the interest of time, we'll get right into some questions from the audience here in our live panel discussion. So, Allison, we'll start with you. We have a question from Paulo in Brazil. It says, what is your opinion to include the microbiome determination at household and work site levels? And if you don't mind, for myself and everyone else here, perhaps give us your definition of what the microbiome means in this case and its connection to environmental exposures.
Yeah, so microbiome being sort of all the bugs that live both inside and outside of us, right? So looking at bacterial and viral and fungal sort of populations, whether that's within the gut, that is an active part of a person's metabolism and directly impacts their health, or whether that's looking sort of within, as the question suggests, their home or workplace for what they're exposed to there. I think it's really exciting and really promising. I mentioned I've working with Stephanie London at NIHS looking at the microbiome of dust samples in an agricultural cohort and looking at health outcomes. I think it's really promising. I think we're still in the infancy of knowing what to do with microbiome data. There's still a lot of technical and statistical challenges in sort of dealing with the structured zeros that come in that space. But I think it's going to be a really hot topic area and an important other ohm to collect that really is an important set of exposures, both internal and external.
Excellent, thank you. All right, so we have a question from Jeff as well, and let's see, I think, Peter, this question would be for you. Is there an agreement on what are the core data for environmental and climate that LPS should capture? And are these standardized? And if not, how do we get there as a community?
Yeah, I think the answer to that is that my view is that it's not that LPS studies need to collect the climate data. It's that LPS study designers need to work with climate scientists at the design stage to make sure that the studies are designed so that they can the health interests can draw on the climate data that is being collected routinely, and in particular work across that disciplinary divide is perhaps too strong a word, to actually make sure that you are that the kind of use you're making of the climate data is to generate metrics that are compatible with what health scientists know about the etiology of the health outcomes in question. So I think LPS studies, don't need to collect climate data. They need to be designed and analyzed in conjunction with climate scientists so that there is a mutual understanding of what the data can and can't tell you. And this could lead to design modifications on either side. For example, it would seem to me that perfectly reasonable that if a new LPS study is being funded by somebody like Wellcome, and they are serious about climate health research at the interface, they ought to fund dedicated meteorological monitoring stations that are in the right place to match up with the health outcome data, right? And then from the other side, if health scientists are going to use existing climate data, then no disrespect, but they really need to understand that typically climate data are not data. They're the results of predictive models, and they come with certain caveats. And therefore, you need to really get behind the superficial published data and really work with the climate scientists to understand what those data can and cannot tell you. So I think there are definitely, it's basically about encouraging the two communities to work together at the start of studies.
Excellent, thank you. And that'll lead into my next question for the both of you. But first, I just want to plug a comment here from Philip, and something I agree with. And he was just pointing out a framework and program in Canada that is perhaps very useful to the IHCC participants, and that's the Canoe. So that link is down in the chat there. So thank you, Philip, for reminding everyone of that great Canadian program as well. So I wanted to ask and start with you, Allison. Being statisticians here, where do you see the as methodologically and computationally, do we have the resources we need to address these complex gene environment questions? Is it a matter of just putting them together with the data resources, or are there methods that need to be further developed so we can actually understand these complex questions?
I mean, you're asking a biostatistician, so I'm going to say we're nowhere near where we need to be on the methods. But I think for sure we know that sort of gene environment interactions are hard to find. The effect sizes are small. It's going to take huge sample sizes. We learned that in GWAS mapping, that sort of the number of samples we need was orders of magnitude over what the field thought 15 years ago. And I think we're going to relearn the same lessons with gene environment interactions, which is why it's so exciting, efforts like IHCC that can assemble that kind of sample size. We also need, as Peter said, there's so many types of modeling that need to come in and different expertise that sort of understanding limits to the geospatial models that are estimating those exposures and putting that in with understanding the genetic modeling assumptions, and then thinking a lot about those correlations in space and time. Sort of connecting back to discussions in the previous session on sort of race, ethnicity, genetic ancestry, structural racism, and how that changes exposures in the built environment, and social stressors and that kind of thing is going to take a lot of different interdisciplinary expertise and pretty sophisticated modeling to really disentangle that complexity. Peter, I don't know if you feel the same way.
Unsurprisingly, I would echo Alison's comment that there's a lot more that could be done with using state-of-the-art statistical methods if there were more statisticians in the field. And certainly in the African setting, one of the things I'm particularly keen on is capacity building, so that more advanced statistical methods can be used in country rather than being farmed out to international agencies. I'd be a bit more optimistic than Alison on sample size in the areas that I work, because I tend to work very much on geographical scale population health problems. And there, spatial correlation is your friend. And we've shown in disease mapping projects with the WHO in the last year that if you compare classical survey sampling methods for estimating area-wide disease prevalence with geospatial statistical methods, you can get a 10-fold increase in efficiency. And it's basically because if something's spatially correlated, then what you measure at one location is informative of what's going on nearby. So I absolutely agree with Alison on the kind of sample size you're going to need to look at mechanistic interactions at the molecular scale. I think for getting population health understanding, I think we can actually do a lot more with the existing typical survey sample sizes we've got by actually investing in sophisticated analytic methods and by bringing in the information that you get from contextual information relevant to the social and the natural environment.
Excellent. Thank you so much. I guess this question is to the organizer. How are we doing on time here? By my watch I have is we have a couple more minutes. Okay, thank you. All right, so let's see here. We have a few more comments or questions coming in. We have one from a comment from Marcel Goldberg in France. Says, in my opinion, the minimum core set of data should be geocoded residential histories and job histories, coded with international standard and classifications. That is a great point, and it's something that Alison and myself have realized as we're trying to expand some of this work from PEGS into the all of us, is that, well, if we're going to base exposures on geospatial, that really it's that residential history or address that is the key connection to then the litany of environmental exposure data. So yeah, that's key there, and it's something that's not a trivial exercise either. Let's see, we have another, I guess, Alison, did you want to comment on that, or?
Yeah, I think those are, I would agree with those as great places to start. History address, and then standard classifications for job histories. I think getting that sort of thing standardized and working with folks in the data portal here with IHCC, I think would be obvious next steps. Certainly not complete information, but great steps forward.
Yeah, and if I could briefly, do be careful though. The best is sometimes the enemy of the good. Depending on the question you're asking, spending all your money on getting detailed life histories is not necessarily the best thing to do. It's probably what Alison needs for the kind of thing she was discussing. But if you're looking at a broader scale population health questions, you may be better off actually going for somewhat less finely resolved spatio-temporal history data in favor of being able to get bigger, more geographically widespread samples to cover the kind of climate scales that are operating, because climate is often a global scale phenomenon, not a micro scale phenomenon.
Yeah.
So it does depend on context, but again, it comes down to designing studies to answer the priority questions. But I am grateful for Marcel's comment, not least because I absolutely agree there should be a core data set within LPS that's collecting georeferenced health outcome data. What my earlier answer was to say that I don't think LPS should be trying to do climate science data collection in itself. It needs to, that's where you need the alliance with other disciplines.
I think that's a great point, and it kind of speaks to some of the challenges of bringing together geospatial exposures. You know, it's important to think about the context of the question, because these things can vary tremendously in their spatial and temporal scales.
Yeah.
You know, and in terms of a more local study that would bring in that information, there is the whole, you know, opening up the Pandora's box of personal protection of information as well. And, for instance, if it's a relatively large cohort, does permission need to be obtained to let people know, hey, we're all of a sudden going to glean your residential history? And that might not have been something they signed up for initially. So we need to be cognizant of that. But that's, an entire discussion unto itself about protecting.
Or indeed personal monitors where people move through exposure space in real time. Yeah.
Yes. Let's see here. We have one more comment right now from Philip in Canada as well. Let's see. He said, perhaps speaking to Allison's point, perhaps the molecular phenotypes, RNA, proteomics, which have larger depths of variability, can support gene by environment by incorporating them into the models while not needing as large a sample size as GWAS. Can you translate that, please?
Yeah, right. Well, if it's something we hope to do in PEGS and other studies by collecting data across omes. We're collecting right now epigenetic data that can help us sort of pull in another level. But I think what Philip's referring to is, right, there is broader variability in sort of typical effect sizes for changes in gene expression than just genetic variation at the SNP.
Sure.
Level. And if you're trying to get mechanism and G by E, when you can layer omics data, can really reveal mechanism and narrow down pathways and that sort of thing very differently than just a GWAS or SNP based.
So not only will we could glean better mechanisms by incorporating multi-omics, we could hopefully require less of a sample size to uncover the gene by environment interactions. That's what you're saying.
Yep.
It sounds like a win-win.
Completely agree.
Okay, so it looks like we are at the end of our time. So I want to thank our speakers, Allison and Peter. Thank you so much for your great presentations. Hopefully everyone's found this helpful. And again, I think, Allison mentioned a few times that we are soliciting the feedback from the larger community about some of these questions in the gene by environment space. So please feel free to get in contact with her and or myself regarding any comments or questions there. And likewise, I believe, Peter gave his information as well. So we'll be in here for the rest of the session as well. So please, feel free to get in contact with us with other questions throughout the rest of the conference. So thank you, everyone.
Thank you.
Thank you.
Welcome back, everybody, from the break. Gives me great pleasure to start off this last session of the IHCC workshop, which I'm sure will be of interest to everybody. It's focused on funding opportunities and to obtain resources for the type of work that all of us want to do. Let me introduce myself. My name is Patrick Tan. I'm from Singapore. I run the Singapore National Precision Medicine Program through an office called Precision Health Research, or PRECISE. I'm very happy to be your moderator for today's session. We have 3 panelists, as shown in the next slide. Let me briefly introduce them. Dr. Bruna Galubadis is from the Wellcome Trust. Jonah Kuhl is from the Chan Zuckerberg Initiative. And Dr. Lee Rongling is from the NIH. And each of them will take about 5 minutes to present some of the opportunities. And we'll then move on to about a half an hour discussion on what it means for you and the IHCC. So without further ado, I'll hand it back to get Bruna to kick us off today. Bruna, over to you.
Thank you very much, and thank you for the opportunity to present to you Wellcome's new funding schemes. So some of you might be aware that Wellcome has a new strategy and that we have reorganized internally to best serve this strategy. Our new mission is to improve health for everyone by funding research, leading policy and advocacy campaigns, and building global partnerships. We will do this by funding discovery research, the schemes that I will present here today, and that is funding research across a wide range of disciplines. We have become broader in terms of the disciplines that we will be funding, and it will be any that has the potential to make important discoveries about life, health, and well-being. We also want to focus in finding solutions to 3 of the most urgent health challenges, and these are mental health, infectious disease, and climate. Next slide, please. That's just to give you an idea of how we are organized now internally. And these are the 2 main departments that when you look for funding, you will be coming in contact with. I'll start with the bottom, which is the research funding. This is the group that manages all our funding operations, and they'll be your first point of contact where you send any research question, any queries you have about the schemes, applications, eligibility, all that sort of thing. Then we have the research programs where we are creating the funding opportunities. One is discovery research, where I'm located, and we're leading on the open schemes that I'll present. But also the other parts of Wellcome that will have funding opportunities that please do bear in mind and be aware that there'll be opportunities much more focused in the area of interest from the 3 health challenges, also from data for science and health. We have Wellcome Cares, not just on the results of the research, but also how we conduct research. So there's a department on research environment that will address research culture, ethics, and researcher-led engagement. Their work will be channeled through the funding opportunities that we will be having. Next slide, please. So I will present you here the open schemes, and these are led by Discovery Research. These are the schemes where you send us your ideas for funding. Next slide, please. So what is Discovery Research? What do we mean? What's the new remit of these schemes? This will cover all the fundamental processes that underpin biology. We want to understand how life works, also the complexities of human health and disease, including clinical and population-based approaches, the developments of methodologies, technologies, and tools. We want to make an emphasis. We want to increase our funding in this area. And just to make clear that this takes a broad base of disciplines. It includes the needs, values, and priorities of the people and communities affected by disease and health disparities. It includes the social, cultural, political, and historical context of human health and disease. Next slide, please. These are very briefly the 3 schemes. Please, if you want to apply for them, there's a lot more information, and you should read the guidance that exists in our website. 3 schemes that address 3 career stages. The early career stage for researchers that have some time on postdoc experience, and they're forming their research identity. By the end of the award, we would expect them to be able to lead their own independent research program, which leads then to the mid-career researcher. That's the Career Development Award that will be for researchers that have the potential to be international research leaders. And then finally, the last scheme, Discovery Awards, are for the established researchers. You can apply to these either as a single PI, a single applicant, or as a team application. These 3 schemes will provide salary for the early career researcher and up to £400,000 in research expenses, and the typical duration of these awards will be 5 years, longer if held part-time. Career Development Awards, you can still ask for your salary if needed, if you don't have established position at the university, at your research organization, or if you are required to bring funding. And for example, applicants from LMIC research organizations can also always ask for their salary. So can cover salary if required and the resources need for your research program. These are longer awards. We heard a lot from the community during our consultation. The bigger and larger awards and longer awards is what it was needed, and we are providing for these. So typically you can ask for up to 8 years. Discovery Awards, these will provide the resources necessary for your research program. We are purposely not giving specific amounts that you can apply for. You should ask for what you need, and then it needs to be justified. We will be looking at the justification. And again, longer awards and larger, usually 8 years. Next slide, please. All 3 schemes will be assessed with the same process, which is a 2-stage process. You will submit a written full application. We have stopped doing a short preliminary application that we had in the old schemes. You submit your full application from the beginning. That will be assessed by a shortlisting committee. This committee will make the recommendations, and those who are invited will come for an interview, which is at the 2nd stage. In between, we'll have the 2 stages. We'll have peer assessment. So only shortlisted applications will be assessed by peer reviewers. And just to point out that this will be a technical review. So it will be sent to specialists in your discipline and in the work you are proposing, and they will assess the research proposal only. For the funding decisions, either the shortlisting or at the interview stage, we will be assessing the 3 main aspects, which is your research proposal in terms of the applicant or the team, the skills and experience, and research environment. Next slide, please. Very briefly, what we will be looking in these new long and bigger proposals. We want to see bold, creative, and high quality. And what do we mean by that? Bold is that it aims to deliver a significant shift in understanding that provides a significant advance over existing methodologies or conceptual frameworks, has the potential to stimulate new and innovative research. And with this, we mean within your discipline, but also beyond, how it can influence and develop other disciplines. We want creative and novel approaches. We want that developments on new concepts, methods, or technologies. If you use existing ideas, how are these combined that they give us something new? We want to, somehow it should be clear what is the before and after the project that you will develop. And they all need to be high quality. That means well designed and feasible, clear, supported by evidence. And if you are applying for a team discovery award, then that the team has all the necessary expertise and skills and the rationale for the team approach that is the best suited for your application. Next slide, please. Happy to answer any questions. And as I said, the first point of contact will be the research funding group, and that's the email that you can contact. Thank you very much.
Okay, hello everyone. It's great to be with you. My name is Jonah Kuhl. I'm a science program officer at the Chan Zuckerberg Initiative and lead our program in single cell biology. So I'm going to give you just a very brief overview to the Chan Zuckerberg Initiative. I suspect that as a relatively young organization, it is perhaps unfamiliar to many of you, and for those of you that have heard about CZI, as I'll refer to it, we also continue to change and update some of our focus areas and opportunities, and so it will be a nice opportunity to update you on our progress and the sorts of funding mechanisms and areas that we're focused on. So let me first start with the overall mission of the Chan Zuckerberg Initiative that is now almost exactly 5 years old. And it was founded by Priscilla Chan, a pediatrician and an educator, and her husband, Mark Zuckerberg, who is a technologist and software developer. And the goal that Priscilla and Mark set out is an overarching mission to support science and technology that will make it possible to cure, prevent, or manage all disease by the end of this century. And the statement here is important and a lofty one, of course, but it's really focused on 2 things. One is catalyzing progress and focused on over the next 10 years on accelerating biomedical medicine by a focus on both basic science research and also building and fueling that research with new tools and technologies that will accelerate the progress, not just against our knowledge of single diseases, but on classes of disease and knowledge more broadly across all disease. And one of the key ways that we're doing this, and I think this is particularly well aligned with the IHCC and your work, is by supporting open models of collaborative research. And so we're supporting, and I'll focus on some of the the exact programs and areas, but really looking at how new technologies and tools can be leveraged by large open communities to catalyze rapid progress. And so with that, maybe the next slide. Great, thank you. So CZI is a slightly different philanthropy than many. So we, of course, fund grants via traditional mechanisms and requests for applications. I'm not going to go over any specific RFAs today, but would encourage you to go to our website, and we have several open, but also open RFAs on a pretty regular basis. And there will certainly be many early in the year coming up in 2022. So these are traditional grant mechanisms. They are open to apply for all of you. In addition to funding RFAs and funding the work in labs around the world, we're also actively building technology tools that are open source and available both to our grantees, but also the community at large. And so we hope that in addition to funding work around the world, we can also fuel and catalyze and help your work proceed more smoothly and make everyone or all scientists better scientists by building strong and robust tools that are open to the community. We collaborate actively, and this is important, as you'll see, I believe, on the next slide, the different programs that we're working on. These are large and ambitious areas, and we do not believe that we're going to solve those alone. And so it's important to us to collaborate with the community, with scientists, but also with other funders. And so many of these programs, we're working with groups such as the Wellcome Trust and others around the world. And then finally, it's really important to us to engage with society, and in particular, to elevate the voice of patients and participants in research. And again, I think this is an area where there's a very tight alignment, and we're really excited by the work that many of you are doing to bring that voice in and to help it fuel and inform and prioritize the research and the need for different technologies. Next slide, please. Okay, so very, very briefly, these are the active programs and kind of domains of research that are ongoing within CZI, and I'll mention them very briefly, but there's much more information on the website, and I, of course, would be happy to connect you with any of the leaders of these different programs or go into them in more detail. So we are supporting a physical research hub here in the San Francisco Bay Area called the Chan Zuckerberg BioHub. It's a physical institute, and its important role is to connect the existing excellent research universities here that are based in the Bay Area, but historically have struggled to collaborate. So these are Stanford, University of California in San Francisco, Medical Center of the UC system, and then UC Berkeley. We have a program focused on imaging and imaging technologies fairly broadly. So that this group is really working on technology development and supporting technology development in areas such as visual proteomics or deep tissue imaging, and as well as building software tools to help with the growing volume and amount of data within these imaging modalities. We have a program focused on neurodegeneration, and in particular, bringing new talent and views into the neurodegeneration field. So it's really a model about a distributed collaborative network and encouraging growing new expertise within that field. A program in open science that is supporting infrastructure and tools that are making it easier to share data and findings more rapidly. So a good example is our support of the preprint servers, bioRxiv and medRxiv. The Science and Society program that I mentioned earlier that is supporting and aiming to give a voice and increase engagement with patients and participants in the research lifecycle. And then finally, the program that I lead, which is focused on single cell biology. So developing tools and technologies at the resolution of a single cell, applying these at scale to both generate references and support for global international collaborations such as the Human Cell Atlas, as well as applying it to disease to try to clarify cellular mechanisms of disease that have previously been interacted with. In all cases, cohorts, data interoperability, data formats are critical to us. And again, I think there's a lot of alignment between the various efforts across the IHCC membership and these programs. And so I would encourage people to learn more or reach out to me if you have questions. Next slide, please. Okay, and the final point just to make is, with the exception of that first program, the Chan Zuckerberg BioHub, CZI and all of our open RFAs are very, very international. So this is just a snapshot of the single cell program and groups that we are currently funding. And I hope what you can appreciate here is that we both receive and welcome applications from around the world, but also support groups from around the world. And maybe this is something that I'll touch on more in the comments, but one thing that we are very happy to do and really look to do is to foster collaborations that connect groups internationally. And we can do this as a philanthropy and with some of the flexibility. And so this is an area where I think we can have a differentiated impact and again, support some of the ongoing work within IHCC to connect different efforts and support the best in global science. Next slide, please. Okay, and with that, I'll thank you. This may be small, happy to share it in the text, but would point people towards our website. I have included my email address as well as my Twitter handle if there's a way that you prefer to reach out. And on the website, there is a way to sign up for updates of all new and current RFAs. So thank you very much.
Hello, everyone. It's my pleasure to give a presentation on the history of NIH support on IHCC, which area have been supported, and the NIH policy for foreign applications, and how to write an NIH application. Next, please. 2015, NIH compiled the information on large cohort programs. The large ones defined as a cohort include at least 100K participants. The information was presented to the heads of international research organization or HEROs. HERO agreed on in 2016 to bring cohorts together to maximize investment by encouraging data sharing to improve effective size, to address questions none of them can answer alone, and to address pressing questions in global health. So NIH and the Wellcome Trust agreed to co-fund this initiative. Next, please. In March 2018, the first cohort summit was held at the Duke University in North Carolina, USA, and 100 individuals attended the summit. Next, please. April 2019, IHCC held its second cohort summit in Iceland. There were 117 representatives from 59 cohorts of 29 countries attended the summit. Next, please. In May 2020, due to the COVID-19 pandemic, the third cohort summit planned to be held in Santiago, Chile, had to be changed to virtual summit. Over 160 attendees from more than 23 countries attended the virtual summit. Next, please. So start from the last year, the virtual cohort member workshop was added as an annual workshop to mainly provide a platform for cohort members to establish meaningful connection and to identify collaborative opportunities. So this year, next please. In May of this year, IHCC had its fourth cohort virtual summit. Now we are at the second virtual cohort member workshop. IHCC also received funding support from, next please. Also received funding support from Dolph's Alzheimer's Collaborative and the Charles Zuckerberg initiative this year. Next, please. What has been supported? The funds have been used to support the secretariat to coordinate the consortium activities and the summit and the activities of following 4 working groups: data interoperability, policy and system, scientific strategy, as well as the training, sharing, and the capacity development. Next, please. You can see from this slides that how many pilots that has been funded through NIH Wellcome Trust and Charles Zuckerberg DAC. So the NIH and the Wellcome has been used to support the 5 projects, and Charles Zuckerberg and DSS support each one of these projects. Next, please. NIH has supported some foreign applications. The 27 NIH institutions and centers providing leadership and financial support to researchers both inside and outside the US. You might find the details information from for submitting foreign grant from the link in this slide. According to the NIH policy, all applications must be undergo peer review process. Next, please. When an investigator plans to submit a foreign application to NIH, the following additional criteria for review must be applied. First, a project has special opportunity for advanced research through use of unusual talent, resources, populations, or environmental conditions in order countries that are not really available in the US, and project has specifically relevant to the mission and objectives of NIH ICES, and has a potential for significant advances in health science in the US. To know more details, please follow the link on this slide. Next, please. How to write an NIH application. NIH has a specific web page to provide the details of writing a successful application. Here, I list the 6 important writing tips. The first, make your project go realistic. Second, be organized and logic. And third, write in clear, concise language. And 4, share your idea on one-pager. And 5, edit yourself, but also enlist help. And then lastly, share for comments. You can also find the sample application from webpage and watch YouTube video by search how to write a successful NIH grant proposal. Here I provide example, that's a YouTube example. You can click on a link to look at the example, how to write an NIH grant. Especially for the sample application, you can find all kinds of mechanisms that the sample applications like U, R, K, or different mechanisms of NIH applications. That is all about my presentation. Thank you for your attention. I'm happy to answer your questions. Also, I list my email address and my web page. If you have any question, please contact me.
Thank you very much. Thank you very much to our 3 speakers, Jonah, Bruna, and Rongling. For people who want to ask questions, obviously, please indicate in the chat. And perhaps I will start off the first question by asking all 3 of you a question, and maybe I can ask you to respond. Given that this is an IHCC workshop, and given the interests of your own funding organizations, are there particularly big questions that you might be particularly interested in, that you see the IHCC as being a very unique platform that's well positioned to address? Maybe I'll ask Bruna to start us off on that question.
Yes. So IHCC offers a ordinary platform for population research and for really maximizing the information that cohorts have by allowing this cross-cohort analysis that allow to answer unique questions that no cohort alone can answer. We've just had in the previous session with the climate and health presentations a perfect, beautiful example of why one cohort alone will not be able to answer questions, because you just need the whole range of exposure that one country or one site alone will not provide. That's an example. Another would be how by doing combining different cohorts, you can study the same association in countries or populations confounding a structure. So actually, by having all these cohorts together, you can go to a step much into the discovery and understanding causality. That's just a couple of examples. I think the ability to cross-cohort analysis is an amazing feature that IHCC facilitates enormously.
Thank you. Let me flip that over to Jonah, because you mentioned the different initiatives of the CZI. How do these best intersect with IHCC in terms of what the initiatives want to do?
Yeah, so I totally agree with the point of how various efforts can come together to synergize, and in particular, synergize on questions that incorporate cohorts, and the global population is something that's incredibly important to us. I think there's another point that I might make, and I think we think about a lot, both in the work that we support, but also our work in trying to build, and that's data interoperability and the challenges that exist there. But I think the IHCC is uniquely positioned to take on some of these challenges and have taken on some of these challenges within large cohort studies. And so this is something that I think is really challenging. It's often one of the major barriers. And so I think this is a very exciting area that is heavily technical, but something that we're very interested in, and really have looked to and inspired by some of the progress and ways that the IHCC has approached this. So data interoperability is actually, in fact, one thing that we think about a lot in connecting those programs, because fundamentally the investigators and groups involved in each, but if the data being generated in the work that I'm focused on in single cell biology is in a format or is not compatible with asking questions in neurodegenerative research, then ultimately the utility is hampered. And so there needs to be flow of ideas and people, but also to support that with the underlying.
And how about WrongLink? Obviously NIH has supported IHCC through the early days. What's the opportunity there that you see for the United States? Because obviously NIH work is focused upon needs of the US.
Yeah, as I presented before, the NIH interest in the specific area that from a foreign country. For the IHCC, I think specifically for IHCC, NIH interested to support the low middle income country that included into the IHCC, as well as the data sharing side. That also important for NIH to support this IHCC to conduct or build the data sharing platform that the global investigators can share this data. And also just a brief that the NIH already, the IHCC already submitted the application, which will be reviewed in this month, the 18th, November 18th. So that one is for the next 5 years support of IHCC. We will see what we can get from this reviewers' critiques. So, and other opportunities within that NIH 27 institute and the centers. So each institute has their specific area that they are interested to fund the foreign application. So that's why I suggested all these IHCC members to look at, based on your research area, look at which institute is appropriate for you to submit application.
I think that's extremely good advice. I'll throw another question out, and anybody that maybe wants to take on, this is more, give me some examples of grants that haven't worked. Obviously a lot of people put in a lot of effort. What are the features of a good application, and what are you not looking for? Anybody want to take a stab at that?
I'm happy to. Go ahead, Bruna.
Yeah, please. I think we want to know, what works, right?
and we'll probably repeat ourselves a bit with this. I think what I said during the presentation, the before and after, this shift in understanding, this step change in knowledge. The applications that don't do well are the ones that add a little piece of information. It can be very valid, very rigorously conducted, very research, but only add a small piece of the puzzle. These are the applications that just are not successful given the high competition that we have. So really look for bold and big questions and innovative ways to answer. I'm just bringing one topic and I'll let my colleagues probably add on to that.
Yeah, so I was going to say something perhaps similar, but we'll try to add to Bruno's example. So for us, we also tend to fund in relatively new domains. And so the projects that we're often looking for, and depending on the different calls are quite successful, are genuinely new, more speculative ideas. And there are opportunities to develop new technologies. There are opportunities to take existing technologies and apply them to different cohorts or populations or diseases for which those technologies have not been applied. And so they can feel at times like maybe they're too preliminary. But we really are looking for that sort of work, genuinely looking for that sort of work. And so I think the projects that often don't land as well or aren't as competitive are, I think, similar to what Bruno was saying, those that maybe do have a really long history or a very clear line and path forward, and our sense is that those are likely to be supported elsewhere. And so kind of those newer, more speculative, less well supported, perhaps we have no requirement for preliminary data, for example. We leave that space open because people often like to support it, but when we send grants to reviewers, we instruct them to not require preliminary data. It's not part of the process to really try to stimulate those ideas. And so I would really encourage people when thinking about projects at CCI to think about what doesn't fit elsewhere. And these might be new ideas, they might be ideas that connect groups around the world. And so I think when thinking about what doesn't fit, it's often that other category.
Thanks very much. We now have some questions from the floor. Particular Scott Sunseth has a question on, I think that's directed to Jonah, but also I think the others can talk about this idea of participant research. So can you expand on this Science in Society program and specifically participant research? And could information gather program from this program help the global cohorts, and how do you learn from different cohorts? I think, and maybe this is something I also like to get Bruna and Ronnie to think about, the role of the participant in engaging people in research. I mean, is that in your mandate? So maybe, Jonah, you want to take a stab at that?
Yeah, I'll try to take a stab at it. It's a great question and fascinating one. So historically, and by historically, I really mean over just the last couple of years at CCI, the Science in Societies program and focus here has been through a program called the Rare as One Network, which is supporting patient-driven advocacy organizations focused on individual rare diseases. And so in this way, it's actually maybe very complementary to the scale and cohorts that the IHCC is thinking about. And it's thinking about trying to lift the voice of patients or family members of patients that are affected by rare disease. Now, I think the real power in these networks is that the needs and the priorities for research from those patients is not always exactly what certainly I would expect as a researcher. And so having that voice be present and including those patients and those advocates in research networks is a really powerful way of understanding not just progress against certain diseases, but kind of prioritize progress through the patient perspective where the impact would be greatest. And I think the same is certainly true at scale. So as the cohorts get larger and different regional, health disparities or, different lifestyle or environmental effects that may not be appreciated. And so really elevating that voice and having it be an active part of the research enterprise is the focus of that.
Thanks, Jonah. Can I ask maybe for Rongling and then after that, Bruna? So how do you see the role of the participant now? For many of these cohorts, we're talking about engaging with human beings. How much of that plays a role now in what you fund in terms of engaging the participants, learning from them?
I think engaged participants is important because this is a global or international consortium. We have participants from all over the country, all over the world. Some of them they have a genetic difference among the population, and also we have environmental difference among the world. So from yesterday and today's presentation, we can see that especially for today's environmental information, we haven't started yet. If we can start that one, we really can bring back some information to local or to individual cohorts. Then, for example, for COVID-19, yesterday we heard about the mental health, that survey. You can see that from that results, you can see some information or some results really benefit the local country, how to the manage of the population or the people who have the COVID-19, how to manage their mental health, which is also important. So the IHCC is not only collect data or collect information from each cohort members, but also bring the benefit to each cohort, bring the data to each cohort, additional generate data to each cohort, and also bring knowledge to each cohort. Another way, and I just want to answer the question, whether they can, each cohort, the local activity, can help the local activity. I think all this benefit or all this advantage of being an IHCC member, you can apply to your local cohort or expand your local cohort, do your own studies that are unique to your country.
Bruna, you want to speak from the point of view from the Wellcome Trust on participants and in this era of engagement?
Yes, definitely. Wellcome has always placed big importance and big relevance into engaging the public and the participants in research. And for the cohorts we've funded, it's been always a big consideration how much the participants were engaged, how much the participants were part of the decision process into what areas the cohort was going to focus, the methods that they were going to use, and there were always big consideration into the funding decision. And either in the Discovery Research Awards that I've presented or any other funding opportunities that might come up in the health challenges, that is always going to be an important component to demonstrate that we take the views of the people to whom the research matters.
Thanks very much. I'm going to ask another question that I think is, and it's highlighted from what Jonas said about some of the tech platforms for data sharing, because there's always this balance between aggregating data from multiple cohorts and enabling multi-cohort versus individual PIs that have invested a lot of work in their individual cohorts. Are there emerging platforms that will permit data sharing in an environment where maybe trust is not 100% there? Jonah, you want to, coming from the founders of the CCI, do you have a sense of what's on the horizon for those sorts of things?
Sure. So maybe just so that I can understand your question a little bit more, Patrick. So thinking about how to promote data sharing in an era and a time where privacy and patient privacy is critical. Is that, I understand that?
I think that's one area. That's actually a very important area. There's also, I think, having participated in some of the earlier IHCC cohorts meetings, there was always this tension between the PIs that invested a lot of work in recruiting the participants, then having the data. And it's a real issue, right? So how to achieve this vision, are there new platforms coming up that enable that sort of sharing to happen? I think all of us gets the idea that this is important.
Yeah, I mean, I think there's maybe a few, and I can speak maybe most specifically to the domain of single cell biology and by extension and to a lesser extent some of the genomics platforms. But I think one particularly exciting path forward is sharing of derived data. And derived data with a very particular lens through particular use cases and utility. So, the raw data, of course, would always be useful, but perhaps is both more onerous to work with, but also, comes with some more sensitivities. And so I do think that there's some exciting examples here. We have been working on one, which is an open platform called Cell by Gene, or pronounced Cell by Gene, spelled CellXGene. And it's a way of sharing single cell expression matrices. So not necessarily the rare data, but it contains all of the information and hard work and expertise that different authors put into that data, including the annotations of all the cell types and metadata, et cetera. So it's really useful. And it kind of lowers the bar for researchers that may be less familiar with doing all of that processing. And I think, similar efforts exist certainly at the European Bioinformatics Institute. I think they're doing a great job, as well as maybe more analysis-focused platforms, such as things like the Terra platform being developed at Google and Broad, that I think, again, are very much through that lens of usefulness and analysis. But I think there's a lot of promise in derived data, not getting too caught up in kind of sharing all data at all times for all people. But we should, of course,
maybe I can ask Bruna and Ronnie, are there ways that the funders can play in order to provide reassurance that, and also to encourage sharing of information and data?
Yeah, no, definitely. And I think there's a lot of emphasis also in open data and data sharing, understanding though that not everything is possible everywhere, and that there's allowing for different formats and different possibilities is probably the best. Agree that sharing the raw data we're moving definitely away, and providing trusted environments where the analysis can go to the data and the data stays, where it's being collected is what gives more trust to people. But again, I think allowing for different modes and different templates is important to make sure that we can bring as inclusive, the more inclusive practices, and we can get people from different geographies the ability to participate and to be able to use other people's data.
Thanks,
Yeah,
Bruna.
I agree with
Ronnie?
Brenna about the different model or different data form that shared among the network. I think for the prospective data or retrospective data, they have the specific informed consent. We cannot share all of the data, but we may share the aggregated data. I think data standardization working group and the policy working group is working specifically on those data sharing. For prospective data, if we collect the future data, maybe we might need to think about the informed consent, what kind of informed consent we can use, since those data collected, then we can have the participants comfortable to share the specific level of data. I think that's both way works.
If I may, just kind of a quick additional note, but I think this is, going back to the last question, also a really important area to engage with participants. We've seen this with rare disease in particular, that the patients are actually incredibly eager to make the data public in many cases, right? And so there is that tension and so there's a real imperative to try to get data out there and get as many people looking at it to make progress against those rare diseases as quickly as possible. That of course is not necessarily true in all domains, but I think it's a really, really important area to engage with patients and participants of how data is shared, and of course, the insights coming back.
I think that's this idea of returning benefit to participants is becoming very strong. We now have another question. This may be the last question that we take because it's addressed again to Jonah, but I think it's more broadly to the others also, that were any of the recently awarded single cell grants actually led by researchers that were in the regions and the communities where the samples were collected? It's addressed to Jonah, but I think we can also ask the same question to the Wellcome Trust and to NIH. So Jonah,
Yeah, so we've recently announced 2 different awards. One focused on pediatric or single cell analysis of pediatric tissues. The other, what we're calling the ancestry networks of human cells. And the ancestry networks are focused entirely on the collection and analysis of underrepresented or tissues from underrepresented communities. The answer to this is a strong and emphatic yes. So actually many are led by researchers and all have researchers as part of those collaborations and networks that are located in Africa, in South America, throughout Asia, India, et cetera. And importantly, it's not just for sample collection and then shipping them for analysis, right? There are active components, and it was actually one of the requirements of the RFA that each team has a community engagement expertise in it. So, participatory research and really engaging with the communities and helping to work with those researchers as well as the local communities. So really important question. I appreciate it.
Thanks. How about Bruna and Rongling?
Yes, I think almost the opposite. We would have to see a good argument for why people from the places where data are collected are not being part of an application, for example, or even if samples are moved out to be analyzed. Again, the justification of why this cannot take place in the country, we will ask these questions. So actually, hopefully, we've moved to a place where if we're not doing these, there needs to be a very strong justification.
Yeah, I would add to Brenna's comments. Yes, that's a single one. You may apply the funding from NIH from different institute or centers for the global network. Yeah, NIH is trying to get the global data and the global study. That's the major support. And that's when I present that the funding opportunity that I mentioned that although NIH and Wellcome Trust now support the IHCC, the individual members in IHCC based on your study, based on your population, then you may search for other individual grant application from specific NIH ICs.
Thanks. So I think the answer from all 3 agencies is that it is a yes, that you will get funded through by being in the country that your samples are being recruited from. Anyway, I think we have now reached the end of our session. It's been a very exciting and illuminating session. I'm sure that all of you will get further inquiries. Without further ado, I think I want to thank Jonah, thank Bruna, thank Rongling for your time, and I will close off this session. Thank you very much.
That was a terrific session, and I want to also extend my thanks to Bruna, Rongling, and Jonah for demystifying some of the funding opportunities that our members might have in approaching all 3 of your organizations in the future, and also for Patrick for leading a really good, being a really good moderator of that session and eliciting some important insights from our panelists. I have the task right now for summarizing this workshop, IHCC Link, which is what we're calling it, over the last couple of days. And I would say one of the things I'm going to try to do is show how we've advanced our strategic plan that Peter so nicely outlined yesterday morning. First of all, it was really impressive yesterday to hear from the pilot project's leaders. Their work has advanced on all fronts. We heard about papers in progress, preliminary data which presages the papers, of course, and funding opportunities, follow-on funding opportunities that were able to be achieved on the basis of the advance of those projects. There was a call from all of them for increased cohort engagement, and I would highly recommend that our cohort leaders, particularly from the low and middle income, less resource settings, really try to engage in those projects. And our our secretariat will be happy to facilitate that, and we will continue to showcase the pilot projects of the IHCC at our future meetings. We also heard about the strategic roadmap from the 4 working groups yesterday, and just to briefly summarize what I heard was that from the training and workforce working group, there will be an opportunity for cohort exchange programs to really help with cross-training from cohorts in different regions of the globe, opportunities to extend a mentorship to particularly our young investigators or junior investigators to learn from the more seasoned cohort leaders. And we, of course, initiated an educational webinar this year, which will continue and already has a great roster of opportunities in the coming months. We also heard from the data and interoperability working group about plans for the Atlas, which is an important foundational platform for the IHCC. The Atlas plans to extend cohort linkages, to increase the level of detail of data that's represented in the Atlas. And of course, you all have received a survey that will help with some of that work. There was a call for inclusion of environmental data, and we heard a lot about the kinds of data during today's session of what could be incorporated into the Atlas going forward. So we heard from the Policy and Systems Working Group about a needs-driven approach to have their efforts really being linked to the projects, to extend their work to crosstalk with other working groups, to continue our focus on data sharing, which is a major challenge for the global community, and also to think about policies about making sure that data that is generated in projects using cohort samples and resources can get returned to those cohorts that generously donated them. This is certainly an important value proposition for cohort participation in multi-cohort projects. And then finally, we heard from the science strategy team about the survey that I mentioned to enhance our understanding of what would be important for cohorts to cohort enhancements that could be done and could be carried out in the future to extend our work with industry as partners, and to work with the Policy and Systems Group on understanding and relieving some of the challenges to carrying out global cross-cohort scientific research. Today, we opened with Nikki Mulder's exciting new data science initiative in Africa, and we spoke about the opportunity of that initiative to link directly to the IHCC in terms of training, open data science and policy. We then had a very, very robust discussion on the inclusion of race and ancestry in cohorts. We learned that an initial survey, not surprisingly, has shown us extreme heterogeneity in the way that race and ancestry and ethnic data are captured. So there's a real opportunity for the IHCC to take a leadership role and contribute to the harmonization of those types of data going forward. We also heard that that there's communication around the types of issues that were raised in that session is a key for engagement, and that researchers need to understand bias in the way that they're capturing data more so than they do today. And this is a very dynamic global field, so we expect that we'll need to both lead in it, but also keep up with it over time. And finally, that social determinants of health, as well as race and ancestry, are inextricably linked and need to be considered together. In the session earlier today on environment and climate data, we heard from the National Institute of Environmental Health Sciences in the United States about a study called PEGS that has really been iconic in collecting survey data as well as integrating global data sets that could influence and that could be relevant to environmental data inclusion, particularly for gene by environment interactions. We also learned that there are significant challenges or at least a significant mismatches, as was stated by Peter Diggle, between the kinds of data that longitudinal population studies are collecting and the environmental data that's out there. So we have an opportunity, I think, again, to play a leadership role in closing that mismatch. we should also recognize that environmental data also harnesses a number of policy issues that we should need to address in terms of particularly privacy and collecting data from personal histories or personal devices. So this is going to be, I think, an important area for us to continue to explore. And as we heard in the last session, it appears that a number of our funders are extremely interested in the incorporation of environmental data into longitudinal cohort studies to learn more about climate and health and environment and health interactions. So I mentioned the last study. I think last session was extremely enlightening and hopefully valuable to our members in terms of learning about how to approach funders, as well as overall for the sustainability of a group like the IACC. So as Peter said yesterday, one of the most important accomplishments I think we made over the last couple of days is building and strengthening our community, thus the moniker IACC Link. And to do that toward our vision of working together to advance science and improving health for all. So with that, I'm going to close this two-day workshop. I want to thank our moderators and speakers for their excellent presentations, insights, and comments. I want to thank all the members that attended the last couple of days. I hope this was valuable to you, and I really encourage you to reach out to myself, Peter, and our secretariat with suggestions for improving how we interact with you in sessions like this or throughout the year. And then lastly, I'd like to thank our funders, Wellcome Trust, National Institutes of Health, Chan Zuckerberg Initiative, and the Davos Alzheimer's Collaborative. And with that, again, thanks for a great couple of days, and we look forward to continued progress for the IACC.